Hello everyone. Welcome back to Dr. Lou's VCE Biological Channel. Today, let's learn together about the 2022 NHT Biology. We will focus on questions related to DNA manipulation techniques and applications. My explanations may need to be revised. If any teachers, parents, or classmates find errors, please feel free to provide your guidance. Step 2. Join the promoter in the coding sequence. DNA ligase does it. Step 3. Cut plasmid. Endonuclease. Step 4. Mix cut plasmid with promoter and coding sequence. DNA ligase. The promoter is a switch for transcription. It turns on the expression of the HGH coding sequence. Since this plasmid contains an ampicillin resistance gene, the cells taking up these recombinant plasmids should survive in ampicillin plates. So, the suspension should be spread onto an ampicillin plate. The cells taking up recombinant plasmid should grow into clones, and other cells without recombinant plasmid will be killed. GM crops are resistant to insects and reduce insecticide usage. In 1990, the insecticide usage attained peak. In 1992, it decreased. So, GM crops were introduced in 1992. The biological implication of GM: the use of only GM crops may reduce diversity and increase crop susceptibility to disease. Ethical implication of GM: large companies may control seed prices, restricting GM crops to wealthier farmers. The TOP1 gene controls the development of embryos. When TOP1 is deactivated, the embryos stop at a very early stage of development. The gene can be deactivated using a gene editing molecule. Breeding with these gene-edited mice resulted in offspring that were all female. No male mice were born as the TOP1 gene was deactivated in the male embryos. The government is considering allowing the TOP1 gene editing to produce only female chickens and prevent the killing of male chickens. The key word is why. That means an explanation of ethical implications. TOP1 gene editing should reduce the number of male chicks that are killed, since this causes pain and suffering to chicks, and reduce the cost of determining the sex of hatched chickens or incubating unwanted chickens, which reduces costs for farmers and increases their quality of life. The components of gene addition are not asked here, but VCE asks the reason that this mouse is considered transgenic or genetically modified. The reasons are transgenic, as there has been an addition of genetic material from another species. Genetically modified, as its DNA is altered. PCR is used to amplify a section of the DNA or RNA in a sample. After completion of the PCR, the DNA can be separated using gel electrophoresis. M lanes are the marker of known DNA sizes, and lanes 1 to 6 are DNA samples. The gel is 3% agarose. Scientists should determine the optimal temperature and timing to carry out each of the stages of PCR. If a strict protocol is determined and followed, the results of PCR is very accurate and reliable. Incorrect results occur in less than 0.0001%. What is PCR? PCR is a polymerase chain reaction. Explain the design primers for a target DNA sequence. The sequence of target DNA strands must be known. The target DNA sequence is unique and can be determined. A complementary strand of signal DNA strand must be synthesized. These primers should attach the target DNA strands specially. Primers can attach to the ends and provide a region for Taq polymerase to join. The shorter the DNA, the faster moving. Lane 1 is the fastest DNA, so it is the shortest DNA. Identify 2 factors that may alter the results seen in the gel and explain them. These results are actual PCR results. This question required students to discuss factors that would affect the process of PCR, not the process of gel electrophoresis. Factor 1. Incorrect temperatures. Explanation. Primers may not have annealed if the temperature was incorrect, or DNA polymerase may have been denatured if the temperature was too high. Factor 2. Timing of each stage. Explanation. If the time was too short for annealing, the primers would not bind, or if the denaturation time was too long, the DNA may degrade. If the extension time was too short, it would be insufficient time for complete DNA replication. These two factors are mentioned in the questions. The temperature and time of each stage should be optimized. In DNA manipulation techniques and applications, 5 multiple-choice questions are worth 5 marks, and 2 short answer questions are worth 15 marks. The overall score for this section is 20 marks. With a total possible score of 120 points, the percentage for this section is 16.7% of the total score.
If you need my help, please add me on WeChat or WhatsApp, and send me an email. Thanks for your attention. Please note Dr. Lou's VCE Physics channel, Dr. Lou's VCE Chemistry channel, and Dr. Lou's VCE Biology channel.